but I know. So we're going to continue on talking about some of the basic chemistry stuff here today. I just want to, and I'll do this occasionally, it's sometimes a fairly easy way to present stuff directly to you in class. I'm just going into the book here, and I just want to scan through a few things to kind of tell you where we are. To begin with, as far as this chapter is concerned, it has these 6 sections. I've basically jumped past the first few sections here about atoms and bonding and reactions, that sort of thing. What we did last time was to look at water, and there's a section about that, and I just want to point a few things out real quick about that. And then we're going to go on to look at the last section mostly today. These 2 sections have almost the exact same title to them, which is inorganic compounds, essentially human functioning, and organic compounds, essentially human functioning. So we'll just make that little distinction here. To begin with, this section just defines the difference between inorganic and organic. And essentially it is, from a chemical standpoint, something's considered organic when it contains carbon. Okay. There are a few exceptions to that, but for the most part, that's what it is, because it's based on how carbon functions as a atom to make particular structures. So I don't exactly start off talking about inorganic versus organic so much as just the idea of small molecules versus larger molecules. And so in covering small molecules last week, not last week, last time on Tuesday, I really just talked about water, and we sort of concentrate on the properties of water. The book addresses the exact same thing, just says in a slightly different way. For example, instead of high heat capacity, which was a term we used last time, it refers to heat sink, or talking about liquid mixtures, that's getting into the universal solvent aspect of it, which is said right here in the text. So the book sort of addressed those same things. In the book where it's talking about water, it introduces this idea here as far as the presentation that I put together for you on Prezi that you watched before Tuesday. I included this particular image from the book with the chemical reaction thing. This isn't so much a property of water aspect as much as just water is sort of involved in a lot of the chemical reactions in our body. So I added that in there. And then it goes on to talk about salts. We did sort of touch on sodium chloride in terms of how it dissolves in water, which is what this picture is kind of getting at. I didn't talk about acids or bases in class, but that was in the presentation. In the book, after talking about acids and bases and pH, it has a section on buffers, which I don't really talk about in A&P 1 a whole lot, just because to talk about it at this point, it would sort of probably get lost in all the other things that you're getting introduced to at this point in the course, and they don't actually come up until A&P 2. They're very important in understanding a number of things about the blood and how the respiratory, digestive, and urinary systems work together, but that's stuff that's quite far off in the future. So setting it up now for a payoff, half a year from now, isn't really going to work too well. So the book mentions them. You should read that section, but I don't really deal with them until NP2. So that moves us to the organic compounds part. And again, it's largely based on the fact that carbon's the root of all of these, and how carbon makes bonds is very important. In the presentation, I talk a little bit about organic compounds. I introduce your sort of very basic hydrocarbon type molecule, and I introduce this one here, which is ethane. It is not the most simple carbon molecule. It's got 2 carbons in it, actually, but it does show how carbons connect together. And in the presentation, I go on and talk about just the idea that you can sort of increase the number of carbons in a molecule like this, and I jump from ethane that has 2 carbons to octane that has 8. I use octane 1 because the word octane, I think, is pretty obvious that it's referring to the 8 carbons in it, with its octo in the word. And then also octane, a lot of people are familiar with that name, that word, because of its relationship to gasoline. But what I want to do now is to introduce, and building up to the idea of macromolecules, and we're going to talk mostly about proteins, the idea of functional groups. I would kind of informally define functional groups as groups of atoms that are associated with each other on a regular basis, that are part of larger molecules, and will often confer particular properties onto that molecule. Now, if I were making this particular table, I would have actually started with the methyl group down here for a couple of reasons. First off, it's a nice way to kind of introduce what I'm doing here with the idea of organic molecules and hydrocarbons. So a methyl group is a carbon with hydrogens attached to it that is part of a larger molecule. What that means really that carbon can make 4 bonds, and so one of those bonds is going to be back to the rest of the molecule, and so the other 3 are hydrogens. The YL ending there is just a chemical way of referring to it as being part of something else, but really what it is it's the molecule methane that's just part of something bigger. Okay, so methane is the sort of simplest hydrocarbon type molecule. Where was you going with that? We can take methanes or methyl groups and put them together to make something larger. So you could kind of think of ethane as 2 methyl groups that are put together. Not too many biochemists will actually think that way, but it's essentially what it is. The other reason why, the first reason why I wanted to kind of put it at the top of the table here is that it's really sort of the basis of building organic molecules. The other reason is that compared to everything else on this table, it's unique or different, however you want to say it, because of the property that we usually pay the most attention to with functional groups, which is how they interact with water. Now, ethane, which I just described as 2 methyl groups, or we just think of it as your basic hydrocarbon kind of molecule. How does it interact with water? How would you describe that? Can you tell me anything about the bonds that are present in that molecule? What type of bonds are they?
Hydrogen bonds?
No. When we draw molecules with lines like that, that's referring to covalent bonds. Okay. Now, the fact that you said hydrogen bonds tells me that you have a misconception a lot of students get because of the unfortunate name for a hydrogen bond. I really wish hydrogen bonds weren't called hydrogen bonds, because students will see something like this and see a bunch of hydrogens bonded to something and think that those are hydrogen bonds. That's not what hydrogen bond really means. Hydrogen bond is when two molecules are attracted to each other kind of weakly, like two water molecules like we were looking at last time. One of the sides of that attraction is usually a hydrogen, and that's where the name comes from. However, it's misleading because not all hydrogens can do that. Okay. Now, so this is a covalent bond. What type of covalent bond is represented in this molecule here?
Nonpolar.
Right, nonpolar. What does that mean?
Equal.
Did you say equal?
Yeah.
We'll work with that because that's the important word. A covalent bond is when two atoms share electrons. And so a nonpolar covalent bond is when they share those electrons equally, which is to say the electron from hydrogen and the electron from carbon that are shared in this bond spend equal amounts of time around hydrogen and carbon. Okay, so let me just represent carbon and hydrogen making a bond. This isn't, there are three other bonds that carbon can make. But hydrogen came to the party with one electron in its orbital shell. Carbon came, let me draw a carbon a little bigger. Carbon had two electrons in its inner shell, but then it has an outer shell with some electrons, and one of those is shared with the one in hydrogen. And really those two electrons now have this sort of complex orbit, and this is not exactly how chemistry works, but it sort of fits with our planetary model. This electron is going to go around hydrogen and around carbon and like that. Okay. And this carbon electron is going to go around carbon and around hydrogen. So they have this kind of figure 8 orbit now. Again, not really how it works, but we're sort of extending the model of the planetary model of the atoms that we've been working with. So in a nonpolar bond, the electrons spend equal times around either one. Okay. In a polar bond, like in water, which would be oxygen is the other one, then the electrons are going to, well, let me just erase these orbits real quick. So the electron from hydrogen is going to spend more time around oxygen and occasionally go around hydrogen. And the one in hydrogen is oxygen is going to stay around oxygen mostly and occasionally go around oxygen. That's the idea of unequal sharing, that in a polar bond, they spend more time around like oxygen. Which means the oxygen having the electron more than the hydrogen is partially negative, and hydrogen not having the electrons as much is partially positive. Okay, so unequal sharing electrons, you have the distribution of charge. For ethane or a hydrocarbon molecule like this, these bonds are nonpolar. The electrons are shared equally between them, so there's no partial charge on either side of that bond. Okay, get it? So if you have a water molecule that's made up of these kinds of bonds and partial charges, and an ethane molecule that's made up of nonpolar bonds, they don't interact because the partial charges in water and the neutral charge of the bond in ethane, they can't interact. There's no attraction between them. Okay. See that? And so this hydrogen, since it's bound to carbon, will never participate in a hydrogen bond because it has no partial charge to interact with something else. So that's the misconception a lot of students get because of that unfortunate name of calling it a hydrogen bond. This hydrogen doesn't make hydrogen bonds because it's a nonpolar bond, because it's part of a nonpolar bond. This hydrogen in a water molecule will, because it has that partial positive charge, because its electron actually spends more time with the oxygen. Okay, everybody clear on that idea? Okay, so back to my original question, which was, how does ethane interact with water? Because it's made up of nonpolar bonds, and any bond between two atoms of the same element is going to be nonpolar, just the rule. So all of the bonds here are nonpolar. If ethane is in water, it's not going to interact with the water molecules at all. Okay, so it's sometimes referred to as hydrophobic, which means that it doesn't dissolve in water. Now, what we can do with functional groups is we can start to add properties to something like ethane by changing some of the elements in here, or some of the atoms in here. So if we take one of the hydrogens off of ethane and put oxygen that's also bound to a hydrogen, that OH, which is called the hydroxyl group, is a polar bond. Okay, so now we have some charge, and I'm not going to say negative or positive out here, but this end of the molecule has some charge. Okay, and so it can now interact with water because water molecules can participate in hydrogen bonds with this out here. Adding that hydroxyl group onto this, what was the ethane molecule, changes this so that it's no longer called ethane. Instead, it's called ethanol, which you've probably heard of. What is ethanol?
It's fuel from corn.
Yeah, unfortunately, nowadays, people usually associate it with gasoline because it's now usually an additive in gasoline. But you're probably more familiar with it, maybe not by name, as the stuff we refer to often as alcohol. Okay, so if you have beer or wine or liquor, what we just refer to as alcohol, actually from a chemical standpoint, is ethanol. So yeast that's used to brew beer or make wine or ferment grains or whatever it's going to be to make liquor. They eat the sugars that are present in the grain or whatever you're making, grapes for wine or whatever. They eat that. They don't have teeth, of course. They digest it. And their waste product is ethanol. Okay. It's just what they give off, like we give off carbon dioxide as a waste product. Well, actually, they give off carbon dioxide too, but they give off ethanol as a waste product, which accumulates in the solution where they're fermenting, and it makes that solution an alcoholic drink. Okay, so that's to say, just anything that you add an OH group to, as far as a chemist is concerned, is called an alcohol. And just because something's called an alcohol doesn't mean that it's something that you're going to drink to get drunk. Probably most things that are classified as alcohols are actually potentially toxic. Okay, so you can actually do this to any length carbon chain. The 2-carbon chain is ethanol, which is what we're used to. But if we started with just a single carbon, if we added an OH group to methane, we'd get methanol. You do not want to drink methanol. And then as we go up into longer and longer chains, the same thing happens. I think 3 carbons is butanol, 4 carbons is, well, butane, and then with an OH group, it's butanol. 4 is propanol, propane, propanol, and then pentane or pentanol, and et cetera, et cetera. You don't want to drink any of those things. They're not good for you. Right? Ethanol has its problems, but we can tolerate that. But then when you get into really different kinds of molecules, there are other things that are technically classified as alcohols because they have one of these on them, but they aren't things you're going to drink to get drunk. Can anybody think of a common substance that you've probably heard of? Its name ends in OL. And when you tell me, I'll point out that it's actually chemically an alcohol. Can you think of something like that?
Tylenol.
Probably so. I mean, again, it's really just a naming convention. I've never actually looked at the molecule that is active in Tylenol. Oh, wait. It's an acetaminophen. I'll have to look at that. That's an interesting thought. It very well could be, but don't try to get drunk on Tylenol. I'll have to wonder what that actually is. But I was going for a more generic kind of thing, not necessarily a generic drug. But I've asked this question every class I've ever covered this stuff with, and sorry?
NyQuil.
Did you say NyQuil?
NyQuil, and that's for cough.
No, that's NyQuil.
NyQuil.
Yeah, that's something else. But anyways, I ask this question. I usually do not get the answer I'm looking for. And then as soon as I say what it is, everybody goes, oh, yeah, it's cholesterol. Yeah. Cholesterol is, in fact, chemically speaking, an alcohol. It's a hydrocarbon that's much more complex than ethane. It has, I can't remember exactly, it's like 33 carbons, and they're not in a chain, they're in a bunch of rings and all that sort of thing. But there is an OH attached to it, which technically makes it an alcohol. Now, by adding this OH to ethane, making ethanol, it confers a little bit of a polar or hydrophilic property to this molecule. And ethanol can dissolve in water. Okay? Ethanol is considered a hydrophilic molecule, and that's hopefully self-evident because you drink alcohol, beer, wine, whatever. It has to be soluble. It has to be able to dissolve in water for it to be in solution like that. However, for longer ones, like octanol, for example, 8 carbons, that doesn't dissolve in water because the polarity of this end of the molecule and the non-polarity of the 8 carbon chain end of the molecule are tipped towards the, hydrophobic side of things more. Okay. Students who take a class called organic chemistry probably, and I actually never took organic chemistry, but that's beside the point. There's a pretty classic laboratory exercise they do where they take a number of different things like methanol, ethanol, hexanol, et cetera, up to certain length of carbon chains, and they see how long the carbon chain has to be before it stops dissolving in water. That's a fairly common organic chemistry kind of lab. And again, I didn't actually take the class, so I'm not sure, but it's usually somewhere in the 4 to 5 carbon range where it stops actually dissolving in water, because the non-polar bonds on this side dislike dissolving in water more than the hydroxyl group on that side does like to dissolve in water. So that's a hydroxyl group. Makes things a little bit easier to dissolve in water. The next one, which is a carboxyl group, is a little bit more than just an OH. It's a carbon double bonded to an oxygen with an OH on it also, this whole thing. Which is not exactly typographically represented in what's in the table, but that's because they're not really trying to show the double bond and all that stuff. But that's what makes up a carboxyl group. When something has a carboxyl group on it, we usually refer to it as an organic acid. So amino acids or fatty acids, those sorts of things, have that thing in them. And they are acids because of properties of that ending. So this is no longer ethanol. And I actually don't know technically what this is called now. No, I have no idea what it's called now. I was going to try to make up a name, but I just don't know. Because while this technically I think would be called a fatty acid with the, well, now 3 carbons in it, fatty acids are usually longer than that, you know, maybe 8, 10, 20 some odd carbons long. And even then, I don't offhand know a whole lot of names of fatty acids. But again, the length of the carbon chain kind of determines if it can dissolve in water. And most fatty acids have pretty long carbon chains and they don't dissolve in water. And you're probably used to interacting with them kind of in a everyday sense, in that they're oils or solid fats like butter or lard or something like that are made up of fatty acids. The amino group is essentially ammonia that's part of a bigger molecule. So amino and ammonia are related words. So I'm going to take this thing that I started off with that was ethane, and now I've got a carboxyl group on it, so I don't know what it's actually called. And I'm going to take one of the carbons off, the one opposite the carboxyl group I just put on, and add to that a nitrogen, which has 2 hydrogens attached to it, so an amino group. Okay. And now what we're looking at is a very, very specific molecule. It is an amino acid, so it has a carboxyl group, which makes it an acid, and has an amino group, so it's an amino acid. All amino acids have this basic structure, which is an amino group on one side and a carboxyl group on the other side, which are both connected to a central carbon. And then, since carbon makes 4 bonds, it has to be bound to 2 other things. One of the things it's bound to is another hydrogen. And then in the 4th position, what I have covered up with my hand right now, varies from amino acid to amino acid. Okay? And these are the amino acids that we use to make proteins that I'm talking about. So the simplest version would just be to have another hydrogen there. The more complex things that that can come into play, you could add a methyl group on that. You could add another methyl group on that. You could add an amino group on that, et cetera, et cetera. You could do all sorts of things. And there are lots of different amino acids. What you'll often see in the book has this, in that spot, you'll see the letter R. R is not an abbreviation for any atom or anything like that. It stands for radical group, which is an old-fashioned name, but the abbreviation sticks. Nowadays, it's called the side group. It's sort of a variable thing that could be in that position. We're going to work with these things I have up front a little later on, but what they are is models of all of the different side groups. So you'll get to see them in a little while to get an idea of what they look like. But I do have a picture here, which comes from the thing that we're going to work with. Okay, so there are 20 amino acids, and so the nitrogen, carbon part in this picture and in the thing that we're going to work with later on is the tube, which doesn't exactly look like a tube in this picture, but the thing down here. And then the side group, what the R stands for, are all of these different things. So the simplest side group is a hydrogen atom, which is right there, and that's the amino acid called glycine. Maybe the most complicated one, complex one, whatever you want to say, is this one down here called tryptophan. It's more obvious in the models, or maybe it's just that the contrast in this picture isn't that great, but it's two rings. So there's a ring right here and a ring right here that come together, which the one with the blue dot in it is a 5-sided ring, and that blue dot is a nitrogen, and then the other one is 6-sided, like that, and that's attached to where the R is. So that's a little bit more complex than just a hydrogen, but whatever can be in that spot can get kind of involved. That's what that particular one is. So this gives you a little bit of a picture of some of the variety of ways that we can arrange these atoms from the functional groups and come up with a bunch of different sort of things. And there's a lot more possibility than just these 20, but since we're going to be working with it, I'm showing you this particular example. The one that I've drawn here on the board, I want to point out, when I draw something like this, or when anybody draws something like this, the corner of each of these rings, so there are 6 around here or 5 around here, represent where there's an atom, and then the line is the bond that it makes with another atom, and the bond, et cetera, et cetera. If we don't write anything into where those corners are, you assume that it's a carbon, because carbons are everywhere. If it's not a carbon, you'll see something written in, like that N that I wrote there is for a nitrogen. And then carbons can make 4 bonds, so I'm going to write it in, although it's not commonly done. This corner right here has a carbon. The 2 lines there are both bonds, so it's got 2 bonds that we can see there. But carbons can make 4, so there has to be 2 more bonds there, which would be 2 hydrogens. And I'm certainly not going to go around and write that in for all of the carbons in this picture, but you can just assume wherever there's a carbon, it's supposed to make 4 bonds, and so if there are not 4 lines drawn to where that carbon is, you just assume that there's a hydrogen. Likewise with the nitrogen, how many bonds can nitrogen make? 3, right? How many are drawn into the picture right now? 2. So it can make 1 more, which would therefore be another hydrogen. This is an amino group, this nitrogen with a hydrogen. Let me just jump back to the book thing here. Where it says amino group, it says NH2. The number of hydrogens in any functional group actually doesn't entirely matter. Here at the end of a amino acid, the amino group has 2 hydrogens. But here in the middle of a ring, it has just 1 hydrogen, because what's more important is how it connects to everything else. So they're both amino groups, even though they have different numbers of hydrogens. It's just the one in the ring structure has to make 2 bonds to be part of the ring, so it only has space for 1 more hydrogen. Okay. And then at the bottom, there's a phosphate group, which if I have a chance, I'll show you a picture of one of those later on. As you move down through the chapter here, it starts introducing the major macromolecules, and I'm just going to say a few things about them. Carbohydrates, what we often refer to as sugars. The 5 molecules represented here are simple sugars, or monosaccharides. The top 3 have 6 carbons each, and the bottom 2 have 5 carbons each. Carbohydrates, as the name suggests, are carbons and water. Carbohydrate, carbon, water. A carbohydrate is going to have a carbon atom, 2 hydrogen atoms, and an oxygen atom in some multiple of that. So that's a ratio of 1 carbon to 1 water molecule's worth of atoms. The name represents that ratio, but it's not entirely an accurate representation of what carbohydrates really are. Now that we understand chemistry a bit more, the old-fashioned name, carbohydrate, is slightly inaccurate. But we stick with it because it's it's sort of the historical one. The three at the top with 6 carbons would therefore have 6 carbons, 12 hydrogens, and 6 oxygens in them. All 3 of them have the exact same 24 atoms in them. What makes them different is how those 24 atoms are arranged in their structure. The middle one you can see is a 5-sided ring as opposed to the other 2, which are 6-sided rings, but they still all have the same number of atoms in them. At the bottom, those 2 5-carbon monosaccharides would therefore be 5 carbons, 10 hydrogens, and 5 oxygens. So it's just that ratio you're going to see all over and over again. The ratio changes as you get into bigger and bigger carbohydrates, but it's based on that. For example, these are the 3 most common disaccharides, which is 2 simple sugars bound together. Sucrose at the top there is table sugar. And then lactose underneath that here is the sugar that's a component of milk. And then there's a third one, which I have to scroll down to see, that's maltose. That's actually what you find in grains that brewer's yeast breaks down to make ethanol. It's sometimes just referred to as malt, but maltose is its proper name. If you take simple sugars like glucose and chain them together until bigger and bigger molecules, you get into these complex carbohydrates or polysaccharides, like these examples of starch molecules. A similar molecule to starch called glycogen. And glycogen is important in anatomy and physiology. This is what we use to store sugar for later use in our liver and our skeletal muscles, or I should say muscles in general. It's kind of like starch, but not exactly. And it's not just humans use it, animals use it for sugar storage in general. And so you can see that they're kind of similar shapes, the bigger starch molecule and glycogen there. Then the very regularly arranged rows of things at the end, that's cellulose, which we more commonly refer to as dietary fiber. Plants who make glucose through photosynthesis use carbohydrates for structural components in the cells, namely the cell walls. We don't, as animals, don't do that because we don't make our own sugar. So all of our structural components are made from proteins. But dietary fiber is really just the structural part of plant cells, one of the structural parts of plant cells. Talks a little bit about what carbohydrates are for. Mainly it's about that they're for energy. We break them down to get energy to carry out a whole bunch of cellular processes. Then it introduced lipids. The most common lipid, as we tend to think of it, is the fat molecule, which is called a triglyceride, which is described there. I talked about fatty acids and longer chains. So here's 2 examples of fatty acids. Fatty acids come in 2 types, saturated and unsaturated, which is just a chemical distinction. Unsaturated fatty acids have a double bond in them. Saturated fatty acids only have single bonds in them. And so that's represented here. You've probably interacted with fatty acids that fall into these 2 categories. You might not necessarily use these names, but you probably are familiar with them. Can anybody give me an example of the common instance, I suppose you could say, of an unsaturated fatty acid? Where do we usually find unsaturated fatty acids? Or, well, we find fatty acids in food, yes. What kind of food would have unsaturated ones?
Dairy.
No.
Like chunkier?
No. If I had asked where you find saturated fats, then that would have in fact been correct. So we usually think of unsaturated ones being in more healthy foods. Can you give me an example?
Almond.
So almonds, there's a lot going on there, but if you were to extract the fatty acids from almond, I suppose there's something called almond oil or something like that. I'm actually not positive. Maybe it's palm oil. Could come out of that. An easier example would be olive oil or corn oil or vegetable oil, canola, those kinds of things. Oils are usually made up of unsaturated fatty acids. What would be a less healthy alternative to one of those kinds of oils?
One that's saturated.
Correct. So what's an example of a source of saturated fats, fatty acids? Sorry?
Butter.
Butter, right. So your answer, dairy, earlier, there's a lot to dairy, but the fats in dairy tend to be saturated fats because they're coming from animal sources, and unsaturated tends to come from plant sources. It's really just a matter of the chemical distinction. Unsaturated has a double bond in it, saturated doesn't. At room temperature, saturated fats tend to be solid, like butter or lard or something like that. And at room temperature, unsaturated ones tend to be in a liquid form, like oils. So you can kind of tell them apart that way. Except that you can kind of, well, that's beside the point. I won't go there. Can anybody give me an example of an exception to the idea that animal fats are solid at room temperature, like I just said? Or animal fats are saturated and bad for you? Anybody got an example of an animal fat that's
Milk.
better for you and liquid at room temperature, and milk is not it, because there's a lot of other stuff in milk. If you remove the fats from milk, you get butter.
Are you thinking fish oil?
Fish oil, exactly. So fish oils are unsaturated, like the more healthful plant-derived ones. So, and there's something about fish living in cold water environments. They need to have unsaturated fats for some reason. So I'm going to talk about phospholipids more next week because they're important for, or I should say, I'm going to talk about them in the presentation for next week. Well, no, I'll talk about them a little bit in class too, because they're very important about cellular structure, but they're mentioned here. In this same section, after at the top there's phospholipid, then it shows you a couple of other types of lipid type molecules. The one in the middle of this picture, which is now at the top of the screen, that's cholesterol, okay? So over here on the side, we have OH. That's what makes it an alcohol. Everything else in this picture is a carbon or a hydrogen. And when carbon binds to carbon or carbon binds to hydrogen, all of that is nonpolar. They're just this little bit of polar stuff. So carbohydrate is not really able to dissolve in water, but it does have that little bit of hydrophilic thing stuck on the side of it. And then this thing these things at the bottom, which are called prostaglandins, they're lipid derived, but they're actually not hydrophobic anymore because they have a number of things in them. So where's my, there we go. So there's a hydroxyl group here, a hydroxyl group here, a hydroxyl group here, and COOH is just another way of making a carboxyl group. So there's some stuff attached to these lipids which make them more hydrophilic. But aside from those things I just pointed out, everything else in those are hydrocarbon bonds. And they are usually made from lipids directly. We won't talk about prostaglandins in AMP1 anymore, but they do come up. They're important for the immune system and the digestive system, so we'll come back to those. There's a little blurb here talking about steroids, which are just hormones that are made from cholesterol, and then they say something about prostaglandins. We're going to do a little bit more with proteins in a second. I'm going to actually skip over this bit to just wrap up what the book is covering. The next section, they start off talking about nucleotides, and this is a nucleotide. It's not really a macromolecule in the sense of carbohydrates, lipids, and proteins being macromolecules. Rather, it's the single unit of the fourth macromolecule, which is called a nucleic acid. But it's worthwhile to stop for a second to think about nucleotides themselves because they're a little bit more complex than a simple sugar or an amino acid or something like that. They're made up of three major parts. There's a 5-sided ring here representing a pentose sugar or a 5-carbon sugar, which is either ribose or deoxyribose, depending on whether this is a nucleotide for RNA or DNA. And then that's connected to a phosphate group, which we see here. And I said, if I had time, I'd show you a phosphate group. This is one, and there's another picture coming up with a few more. But it's a phosphorus with some oxygens attached to it, which is then attached to the sugar. Because of how the bonds are made within the phosphate group, there's 2 negative charges to it. And in the way that it's usually depicted like this, that's because this oxygen here only makes 1 bond when it's supposed to make 2, and this one here only makes 1 bond when it's supposed to make 2. So they both have like an extra electron that's not participating in a bond. So that's the negative charge that you see there. But it just makes the phosphate group and therefore the nucleotide negative. And then there's a ring structure off to the side called a nitrogenous base. Nitrogenous refers to the fact that they have nitrogens in them, sometimes a couple or few. There are 5 different types of nitrogenous bases, uracil, thymine, cytosine, adenine, and guanine. The 2 at the bottom here with the double ring structure are called purines. The 3 at the top with the single ring structure are called pyrimidines. Thymine and uracil are pointed out here as being slightly different because thymine is only found in nucleotides for DNA and uracil is only found in nucleotides for RNA. Okay. I want to point out a little error in the book down here, which I'm kind of surprised nobody caught. Deoxyribose and ribose are the names of these 2 sugars. And the deoxy in deoxyribose just means that at this position right here, there's just a hydrogen instead of an oxygen and a hydrogen. So it's been deoxygenated, if you will. And the D in deoxyribose is what the D in DNA stands for. Ribose is found in RNA. So this should say RNA, not DNA. A little weird typo there that never got caught, but that's what that is. Those are nucleotides. Nucleic acids are chains of nucleotides, polymers of nucleotides. So we have DNA, which is made out of nucleotides with deoxyribose sugar, and RNA, which is made up of nucleotides with just plain old ribose sugar. And then the uracil is found only in RNA and thymine is found only in DNA. The other 3 are found in both, which if you were to study DNA and RNA in some detail, there's a very important point to be made there, but we're not going to go into that. This is a schematic representation of what a DNA molecule looks like. It's a double-stranded or double helix molecule. A helix is a corkscrew shape, so there's sort of 2 ribbons that are corkscrewed around each other. And the ribbon in this picture, this part here, represents the phosphate and deoxyribose polymer, and then sticking off of each deoxyribose is the nitrogenous base, here just represented by different colored bars. And so that's sort of the picture of what DNA looks like. It's kind of like a twisted ladder or spiral ladder looking thing. RNA has just a single strand, so it looks like half of this kind of, but and it doesn't have a helix shape to it. The helix shape comes from the deoxy part of deoxyribose, giving it a sort of a little spin or kink in it. But that's a nucleic acid. And then finally in this section, I just want to point this out, going back to nucleotides. There's a particular nucleotide that we're going to run into a whole lot in this course.
and it's called adenosine triphosphate. It has an adenine base and a ribose sugar. Those two together are what are referred to as adenosine, which is why it's not called adenine triphosphate. And then it has phosphate groups attached to it. So we already saw that nucleotides have a phosphate group on to them. If we add a couple more phosphate groups onto the adenosine nucleotide, we get adenosine triphosphate. Okay? There's a lot of energy available for use in our bodies in the bond between the second and third phosphate. So when we break that, we release a lot of energy that we can use. So we'll hear about ATP as being a very important source of energy in our cells. When it only has two phosphates, it's called ADP. When it only has one phosphate, it's called AMP. So the MD or T is just referring to how many phosphates there are in that particular molecule. But this is a nucleotide, so it falls into that group of molecules, but it sort of it's very important all by itself, and we're going to talk about it over and over again through A and P, 1 and 2. It'll mostly come up this semester when we get to the muscles and then the nervous system, but then it'll be very important in A and P 2 also, where we talk about a lot of physiology. Okay, so that's the end of that section of the book. I skipped over proteins, so I want to go back to that. I already talked about this, but amino acids are the basis of proteins, and here's the book's drawing of what I drew up on the board, and it's exactly the same thing, except that it looks like, you know, balls and sticks making a model rather than what I drew on the board. But that's the same thing. This picture shows how two amino acids get bonded together. So here we have two amino acids. Here, for some reason, instead of putting an R, they just left an H, which would be fine, except that in the next one, they've put an R in there. So don't get thrown off by that. They're just, what's important is the water molecules worth of atoms here get removed, and so the carbon from the carboxyl group in one amino acid gets bonded directly to the nitrogen in the amino group of another one. And that connection, carbon to nitrogen, is called a peptide bond. Two amino acids that are linked together would be called a dipeptide, referring to the peptide bond holding them together. Three amino acids together would be a tripeptide. We don't really count higher than that. We just go directly to anything more than three. We just refer to as a polypeptide, which in some senses is synonymous with protein. Proteins are usually polypeptide chains. Sometimes proteins can be made up of multiple chains, so they get a little bit more complex. But some proteins are just a chain of polypeptides. What's very important about understanding proteins is how they take on a shape. And this figure here addresses that, which I'll talk about in a little bit more detail in a little bit. But at the top here, there's just a chain of amino acid representing things linked together. That's a polypeptide chain. It's a very short polypeptide chain. Real proteins are going to be much longer than that, but just for the sake of illustration here, it's kind of short. The order that amino acids are found in any particular protein is established by your genetic information, but here they're just sort of generically numbered. And it's showing how that single straight line of peptide bonded amino acids can end up taking on different shapes because of how they interact together. So what I want to do as a little bit of an exercise is to give everybody a little bit of a chance to understand how proteins fold and how they take on three-dimensional shape. So I have three of these kits here. I was showing you the picture from them earlier just to show you the variety of
side chain looking groups. So I'm going to hand out one of these circles and the accompanying tube thing, bendy thing. And I'm just going to put, usually I do this in lab where we have like a continuous row, but obviously there's an aisle down the middle. So you guys are going to be one group, you guys are going to be one group, and you guys are going to be one group. So I'll just put these out on the one side of the room. You guys move over and work together. Over here already, so what I want you to do first, as a group, so everybody gets to do a little bit of this, is to take the side chains that have clips on them and put them on the, this is called a tuber for weird reasons that I'm not going to get into. And the clip is meant to just hold on to it. Now, the plastic clips, over the couple of years that I've had these, have gotten a little loose, so they don't really hold onto the tuber perfectly, but they should kind of fit on there, and if you don't mishandle it too much, they won't be falling off much. So what I want you to do is to take those clips and put them on the tuber just randomly. So everybody can grab a couple and put them on. They need to be about 3 inches apart from each other, so you will not use all of the clips. That's okay, there'll be some left over, but you need a few inches between them to go on to the next part. So go ahead and do that, and then I'll tell you what to do next. Okay, everybody's gotten a chance to do that. I was trying to find something to put on the screen for those unfortunate people that aren't actually participating in this. But I'm just going to leave this up on the screen, which will be kind of boring and people just hear me explain what to do here. But I wanted to capture this for people that aren't here. Okay, now, I did not explain this beforehand because I didn't want you to have any, too much preconceived notion about these clips. You hopefully notice that they have a color coding to them. And on the paper that you saw them attached, it tells you what those colors actually mean. Essentially, they fall into two broad categories, the hydrophobic and the hydrophilic ones. What does hydrophobic mean?
Doesn't like water.
Yeah, we can also call that nonpolar, which really means for them that they don't interact with water or dissolve in water very well. Now, they're all dissolved in water. It's just they're not, the hydrophobic ones would rather not have to interact with the water so much. And hydrophilic would be the other side, which are happy with the water. Now, in the hydrophobic side, there are two clips that are green, which you may or may not have used on your thing. It doesn't matter. If you put both of them on, the first rule to bending up this protein is to bring the two green ones together. So let's say this is your protein, and you have a green clip here and a green clip here. There might be clips in between them or whatever. Just fold that over so two greens come together. Okay. Now that's the first step, so it's obviously a very simple fold. But as you do other foldings, if these end up getting kind of moved away from each other, you can spin the clips around so that they meet up again if the folds end up getting more complex than that.
So that's the first thing you want to do. Let me explain all of this before you start folding it. So start with the green clips. If you have two of them, they come together. If you don't have both of them on there, the green clip will from here on out just be treated like any of the yellow clips, which is going to be the last I give you. I don't want to sit on the table with the mic because it makes noises. They don't even mind that. The next thing is in the hydrophilic section, there are two colored circles, the blue and the red. Blue is for basic and red is for acidic. Now, the color coding there is actually not just because blue and basic both start the same letter. Blue refers to nitrogen, and nitrogen, when it's in a ring structure like, or not a ring structure, but when in one of these structures like arginine here, it has a pair of electrons in its valence shell that are not part of a bond. And so hydrogens that are part of a water molecule or some other, hydroxyl group somewhere are attracted to them. And that's where a hydrogen bonds kind of form, okay? Aside from hydrogen bonds, they'll also bond to hydrogen ions. And when a hydrogen ion is attracted to a nitrogen, it gives that nitrogen and the compound that it belongs to a positive charge. So basic is blue for the fact that nitrogen is involved and they have a positive charge. The negative charge of acids is red because an acid is just a molecule that has lost a hydrogen ion. And those hydrogen ions are usually going to come from hydroxyl groups. So if you take a hydrogen ion off of a hydroxyl group, what's left over?
The acid.
Well, specifically of the hydroxyl group, what's left over?
Carbon and oxygen.
Just oxygen. Hydroxyl group is just OH. Okay. You were probably going to the carboxyl group.
Yeah.
But yeah. Yes, I can certainly see your why you said that, because I talked about organic acids and everything, but I'm talking just about the hydroxyl group. And so if you take the hydrogen ion off and all that's left is oxygen, oxygen is colored red, that's why the acidic one is red. And taking that hydrogen ion off, what's left behind is the electron that used to be from that hydrogen, and that gives the molecule of negative charge there. What do positive and negative charges do together?
Attract.
Sorry?
Attract each other.
Yeah, they're attracted to each other. So after you've dealt with the greens, the next thing you want to do is a red needs to be come together with a blue. If you have both reds and both blues on your chain, and please hold off on doing any folding until I've given all of the instructions. If you have two reds and two blues, you do not necessarily have to bend them so that the closest ones come together. Okay, so if you have a red and a blue at this end and a red and a blue at this end, you could bend it over so the red and blue here come together, and so the other red and blue come together too. You can be inventive in how they bend up, because we're not following all of the rules perfectly. So bring the greens together, then bring the reds and blues together. If you have more reds than blues, or more blues than reds on your tube, and there's nothing for them to pair up with, then the red or blue ends up being treated like a white clip from here on out. Okay. And so let me explain to you what the white and yellow clips are for. They do not get paired up together. What it is the yellow clips, being hydrophobic, don't want to be around water. And the white clips, being hydrophilic, are happy at water. So for your protein, however it might end up getting folded up, you need to have the yellow clip things on the inside of whatever shape you end up getting, and the white clips on the outside. Ideally, if you have a yellow clip on the inside, there should be a white clip near it, sort of blocking water from getting to the yellow clip. Okay. Now, it's very important when you're doing this, and if I don't give this instruction, everybody will do what I'm about to tell you to do. Consider that this has to be three-dimensional. So if you fold something up, and this is a little bit more three-dimensional than most people do, keep in mind that water is coming from the bottom or the top of whatever you're doing. So if you put a yellow thing right here, it's exposed to water here. So you need to find a white clip and fold it over and block it. Okay? So that's going to force you to do something very three-dimensional with your tube. The clips can spin around, so if when you're folding things, you can't get things in the right direction, the way that you have them, maybe all sticking up the same way. It's okay if the clip ends up spinning around the tube in any direction. That would actually kind of happen for amino acids. So go ahead and follow those rules. Green to green, then blue to red, and red to blue, and then yellow on the inside and white on the outside. Follow those rules and fold your thing up however it is. And yes, these things fall off easily. Just keep track of them.
We got to pose together.
Okay. So y'all, like, the only thing we had to do before you do the other
Yeah,
part
was
was
it?
not like.
Right, so like you only have one different alternative.
Yellows.
You only have one of each. So now you have to fold it so that the yellows are on the inside and whites are on the outside.
Yep.
Good luck.
Quite wonderful in concept. Okay. Okay. I got a block.
So I was talking about the spinning thing. So it's going to be out. And then you can. Never mind. Except those two things, since they come together, collectively. So then you have these. Going all right. So these are exposed to water. You haven't turned in. Nothing blocking
No, I guess this yellow is on the inside now, so it's white.
Oh, and those are the only yellow. So that actually
It's
makes
white
it
to
pretty
protect
easy.
the green. This yellow.
We're working on it, so zero.
This one down?
And we might find, like, actually, we get that right now. But now
What'd you put in there?
No.
There we go.
right, so you.
Well, I do have to.
It was at one point.
So which one would you like that? And that blue and that red. And then that might actually be. So you just
The blues are white right now.
just sort of hold them in. Which you can
Yeah.
take them from the sides, the actions inside. And
So you have like this, right? But the power like
no,
that.
no, actually, no. This one's so you kind of get.
Okay, well, it's still outside.
Oh, no.
So can I?
That's not. Oh, that's.
Oh, I forgot.
I got like a 2.
So how do you contain it inside?
It takes a couple of
If I give them an average.
Now, you've all gotten to a fairly decent place. You could probably go a little further. But the point here is to show you how this kind of happens. There's a couple of aspects of this exercise that are very unlike the real situation, which are that you randomly put the clips in order, and in real life, the clips or the amino acid order is established by your genes. If there were a protein with this kind of order that's very hard to fold and doesn't really work, it probably wouldn't be a very successful protein. It probably wouldn't remain very long in your genetic complement or in whatever organism's genetic complement. It just wouldn't be a very good protein to have. So proteins get sort of tested as to how well they do their job over successive generations, and the ones that are good at what they're supposed to do, that are suited to their job, are going to survive, and the ones that don't won't. And survive implies living, and I'm just talking about the chance that the organism survives with them and passes it on to their offspring. So likely the particular sequence that you put together here isn't likely to happen. The other thing is that these are very short. So there's 20 or some odd amino acids there. Actually, I think there's more because there's like 2 histidines. But even a 22 amino acid long chain is unlikely. There are some very short ones, but they're also not randomly ordered. So they fold perfectly being really compact. But a random strand of amino acids this short is very, very hard to actually fold up properly. What I hope you get out of this is how following those kind of rules does impose a particular 3-dimensional structure to proteins. Now, I want to say a few things. I would appreciate it if you, while I'm doing this, kind of help clean up. The board up there has, if you can see it, the exact name of each of the side groups. If you could try to put them back on the circle where they belong. If you can't read that up there and it's taking too long, you want to get out of here in a couple of minutes when I'm done, I understand, but I would appreciate it if you could try to put them back where they belong. In the book, the figure about protein structure. Okay. The primary structure, the just simple line of amino acids, that's the sequence that's determined by genes, and they're linked together as peptide bonds, which is a covalent bond. Then everything else, except for where the cysteines come together, which is actually another covalent bond,
Everything else is hydrogen bonds. So where things start to come together and they're attracted to each other, they make local shapes. The two common ones that are usually presented are a helix and a sheet. A helix is a corkscrew shape. A sheet would be a flat shape. And then if you think about all of those shapes that are present in the whole polypeptide chain, we move from the secondary structure to the tertiary structure. And then if we put multiple polypeptide chains together to make a big complex protein, that's quaternary structure. The dotted lines that we see in the secondary structures are hydrogen bonds. They don't have dotted lines in the tertiary quaternary structure, but those are still being held together mostly by hydrogen bonds. Hydrogen bonds are very weak attractive forces, and you can break them two ways, either with heat or with a change in pH. And by doing that, you can change the shape of proteins. You have probably all done this at least once in your life or had it done for you. Anybody have eggs for breakfast this morning? Anybody else? Anybody had eggs for breakfast ever? Okay. I figured everybody had experience with eggs. The white part of the egg, when it's raw and not cooked, isn't actually white, it's clear. That is a very proteinaceous solution. And when you put it on a frying pan or when you hard boil an egg or something like that, it becomes the kind of rubbery consistency, not really rubber, but the opaque whiteness that we call the white of the egg. And that's because the proteins in that solution, because of the heat, they lose their shape and then they form into a different shape randomly, which gives that part of the egg a very different consistency. You can do the same sort of thing with an acid, for instance, not for eggs usually, although if you want to, try it. More commonly for acid, if you're making cheese, milk proteins, when you expose them to an acid, denature, and then you make the cheese, which has sort of a curd cottage cheese thing. That's really the denatured proteins in milk. So there's a couple of examples of denaturation there. I am holding you over a little bit, so let me end there. I just wanted to say that and explain a little bit about the shapes of proteins here. So next week we're going to start up with the cell stuff. I will be posting a presentation this weekend that you need to watch before Tuesday so that you can be up to speed on what we're doing next week. So thank you and have a nice weekend. Oh, and I see everybody on Monday anyways.
So we have lab every Monday?